Hello everyone, today we're going to be going over Chapter 10, Bio, Photosynthesis. So I like to characterize photosynthesis into two parts. The first one is going to be the light reactions and the Calvin cycle, or dark reactions. Okay, so we start off with the light reactions. The light reactions occur in the thylakoids of the chloroplasts, which are the little baby green things. Okay, so this occurs in the membranes of the thylakoids. So what happens in the light reactions is it uses the Photosystem II and Photosystem I and many different types of proteins in the membrane to split H2O and release oxygen. So if we want to start off by just drawing this thylakoid membrane area, so let's say that this on the outside is the stroma and this is the thylakoid space. Okay. So we start off inside the thylakoid space with water and utilize, okay, I'm going to draw all of the proteins first. This is Photosystem II. And then over here, I'll draw Photosystem I. Not sure why they go like that. Don't ask me. I didn't invent photosynthesis. And then down here, we have an ATP synthase. We'll go over that later. So we start off with the water in here donating an electron to Photosystem I. And then light energy from the sun, remember this is in plants, excites the electron and allows it to be passed on down the chain. This excited electron leaves the water and after that produces some O2 and some H+. Well, two of them. Okay, so here we are building our H+ concentration gradient just from one reaction. There's another little protein here called the cytochrome complex, and this is another way of pumping the H+ ions in from the stroma. So the excited electron is passed down the chain through some various proteins and stuff that I don't really know what they mean or have to know what they mean. And then it is passed on to Photosystem II, where it is excited again, and now we have another excited electron ready to be used. And again, this is from the light energy from the sun hitting Photosystem I. So then you take your excited electron and then move it to another enzyme called NADP plus reductase. Basically, it makes NADP plus into NADP plus, NADPH. So basically it takes some H plus from the stroma and converts it into NADPH, which is used in our next step. Okay, but now we also have to worry about our hydrogen ion concentration being built up inside the thylakoid space. We use this very special protein called ATP Synthase, which uses something called proton motive force to turn a little turnstile in here, or something like that, to pump the H plus in and then pump it out. But with this, also producing ATP from ADP and a free phosphate, inorganic phosphate. So now we've created ATP and NADH, which will be used in the next part of our step called the Calvin cycle. Okay, so moving down to the Calvin cycle. You can break up the Calvin cycle into 3 steps. The first step is going to be carbon fixation. Second step is reduction. And third step is regeneration of CO2 acceptor. So we're going to go over this in more detail in just a sec. So we start off with a sugar called rubisco. Now this is a cycle, so I guess I can draw it in a cyclic fashion. So we will start off with our rubisco and our CO2. The Calvin cycle uses some processes that are a little too advanced we don't really need to worry about for bio right now. All you need to know is that it uses 6 ATPs and 6 NADPHs go into the cycle to produce our 3 G3Ps. And then obviously comes out as NADP plus and ADP. So then we take our 3 G3Ps, and unfortunately only 1 G3P comes out of it, and the other 5 go back in the cycle. Then we use another 3 ATP to get it back into rubisco and ready to continue the cycle. This is Calvin cycle. So we use our 1 G3P molecule times 2 to yield 1 glucose. One of the big things that you should know about the Calvin cycle is that it is very inefficient. So you can see that we used 3 rubiscos to make 1 G3P, so that means we would need 6 rubisco to only make 1 glucose. And it also uses quite a lot of ATP and NADPH. So this brings us to kind of our final point of photosynthesis, where we can go back to the chloroplast. And over here you have your thylakoids, and then over here is the stroma. I'm just dividing it because that's how it's easy to look at, but the thylakoids would be spread out everywhere. So pretty much the light reactions are done in the thylakoid membranes, and they create ATP and NADPH. And it uses light from the sun as well as water. However, the Calvin cycle, Calvin cycle uses the ATP and NADPH to create ADP and NADP plus with carbon dioxide. However, the big difference is that in the light reactions, we produce oxygen, and in the Calvin cycle, we produce the sugar, which is the G3Ps. So as you can see, this is a coupled reaction, where we use the energy from the light reactions in the Calvin cycle. So I hope you guys learned a few things about photosynthesis today. In the next lesson, I'll be going over DNA and DNA replication.